HSADL, High Security Animal Disease Laboratory, is a premier facility of the nation working under the auspices of Indian Veterinary Research Institute IVRI of Indian Council of Agriculture Research ICAR. HSADL is enlisted as one of the few biosafety level 3+ facilities present around the globe and the only one in South Asia at present. This laboratory was constructed by National Dairy Development Board. The primary mandates of HSADL are building national facility and developing competence for handling high-risk animal pathogens, transboundary animal diseases, genetically engineered organisms through basic and strategic research, creation of data bank, transfer of technology providing training and education, and development of biosafety and biosecurity protocols against exotic or emerging diseases of livestock and poultry. The 3-floor design of HSADL is based on the model of BSL-4 lab at Lelystad, Holland. The first floor accommodates the facility for air handling. The ground floor is the working laboratory and the animal wing. The basement contains the decontamination facilities, including effluent treatment plant. There are primary containment barriers. These provide a physical barrier between the worker and the environment and the hazardous material. Apart from these, there are the secondary barriers that delineate the laboratory from non-laboratory areas or from the outside and prevent the possibility of escape of pathogens to the exterior through man, material, air, and water. Entry into the containment lab is restricted. After verification, one can enter the containment complex through designated shower areas. The person then removes all clothes and accessories and enters the interchange area where he puts on the designated laboratory dress. To exit from the containment area, after working, the person has to follow the 3-shower 5-minute cycle. Biosafety staff monitors activities like entry and exit procedure, movement of man and material, as well as communication outside the lab through phone or fax. The research laboratories of HSADL have separate facilities for disease diagnosis, immunology, pathology, biochemistry, vesicular disease, molecular biology, radioisotope handling, cell culture work, recombinant DNA laboratory, and central instrumentation facility. Cell culture is carried out in the cell culture laboratory. All infectious material are handled in the infection room. Maximum personal protection measures need to be adhered to in this room, and highest level of discipline in maintaining laboratory safety and personal safety has to be maintained in this area. As per the biosafety norms, first aid facilities for treatment and over-the-counter medicines are always available inside the containment area. Canteen facilities are also available for persons working in the containment area. The movement of material in and out of this laboratory is closely monitored and scrutinized as per biosecurity protocols. Heavy equipments and other materials are brought in or taken out through the airlock. The airlock is fumigated with formaldehyde for 16 hours. A small dunk tank exists at the blind end of the non-contaminated corridor for taking out of the small materials. The laboratory is also equipped with two double-door barrier autoclaves. One end of these autoclaves is located inside the containment laboratory, while the other end opens outside the containment area. The autoclave discard is then either incinerated in the incinerator or dumped into disposal pit. ETO is a double-door barrier sterilizer that opens outside the containment area. All the material that are sensitive to chemical and heat treatment are sent outside the laboratory through ETO sterilization. The animal pass-through cabinet is the barrier between the laboratory wing and the animal wing and is used for transport of material from the animal wing to the laboratory wing and vice versa. In a dedicated building outside the containment area is the transmission electron microscope facility for ultrastructural studies on pathogens and other molecules. For quick confirmation of various exotic and emerging viral infections of animals. HSADL has a systematically organized animal housing and experimental facility. The receiving shed is an enclosed housing facility to keep newly procured animals in quarantine for 28 days. Animals from receiving shed are shifted to the holding shed after completion of quarantine period and kept here until commencement of experimental trials. The animal wing portion of the containment laboratory is a biosafety level 3+ containment facility with graded negative pressure from -5 to -20 mm water columns. Inside the animal wing comprises of 4 large animal rooms, 3 small animal rooms, and 3 rooms designated for laboratory animals. A separate isolator room is designated for testing the birds and laboratory animals with highly infectious agents or recombinant organisms. The central portion of the animal wing is the post-mortem area. The opening of the rendering cooker is provided here. A cold room facility at -10 degrees centigrade is available for storage of carcass. There is also a specific pathogen-free SPF facility in a separate building. It has 2 super isolators which are constantly maintained at a positive pressure. Birds are reared on autoclaved feed, filtered water and HEPA-filtered air. The flock is regularly tested for 23 different avian diseases. For experimentation, these birds are transported to the containment animal wing in small portable transport isolators. HSADL is an unique amalgamation of biological expertise and specialized engineering applications. The air handling system of this entire laboratory area is administered with the help of 23 AHUs. The air supply to the laboratory is carried out via pre-filters. The exhaust air from the laboratory is carried through the pre-filter followed by the HEPA filters. Depending on the necessity, the negative pressures of the room's areas shall be regulated. Sewage from the laboratories and the animal wings pass through the pipes and sterilized in the effluent treatment plants. Permission for evacuation of the effluent to the outside is permitted only after evaluation of the sterilization report. The data of AHUs and the ETP are supervised from the control room. The data is checked thoroughly at least once a week.
Welcome to HSADL. HSADL is a unique combination of the structural engineering components, and it was established with the financial support from FAO, UNDP, ICAR and World Bank. The lab has contributed a lot to the nation, and by now we have checked entry of a number of exotic diseases at the very entry point, and that is the quarantine. Similarly, the timely diagnosis in the outbreak of 2006 of bird flu disease is one of the major achievement, as this laboratory diagnosed very promptly this disease following the international norms. Considering this contribution and achievement of this laboratory, the World Apex Body for Animal Health, that is designated as OIE, have recognized our laboratory as one of the reference laboratories for the diagnosis of avian influenza. Besides work on the bird flu, we are also working on Nipah virus, we are also working on swine influenza H1N1 and zoonotic aspect of the avian influenza. The achievements of HSADL also include recognition or existence of number of animal diseases in our country, as well as non-existence of a number of diseases in the country. ICAR considered IVRI as Best Institution Award in view of the achievements of the HSADL in the year 2009-10. The scientist of this lab has been conferred with number of awards at international level. Our linkages are with FAO, WHO, OIE, SCPRL Athens, CDC Atlanta and other laboratories of the world. At national level, we are having very good relations and collaboration with the ICMR, DBT, DST, DRDO and NABARD, state departments and many other colleges and universities of the country. Among the neighboring countries, we are having very good understanding for processing of their sample as well as for training of their laboratory officials. HSADL looking for the upgradation of its laboratory to meet out BSL 4 standards as per the international norms. We are also in the process of establishing a BSL 2 laboratory. Attempts are going on to have the OIE reference status for new disease, and that is BVD. Thank you.